Hi everyone, Mrs. Below here. In this video, I'm going to be going over the cellular respiration or cell resp class review. This is just an overview of everything we've talked about in the cellular respiration part of this unit. So the first part here, we have our mitochondria. It's made up of two membranes or two layers. You can see the yellow and the blue. The yellow is the outer layer or membrane, and the blue is the inner layer or membrane. Remember, the outside of the mitochondria, because it's inside of a cell, is the cytoplasm or the cytosol. They're basically the same thing. And that is actually where glycolysis and fermentation take place. So glycolysis is the first step in cellular respiration, and that actually happens outside of the mitochondria. And then fermentation, that will only take place if there is no oxygen present, and that would still be outside of the mitochondria. The Krebs cycle, which is the second step in cellular respiration after glycolysis, and that happens in what we call the mitochondrial matrix, which is the most interior part of the mitochondria. There's all these folds with the inner membrane. And so that space, the inside space here of the mitochondria, that is the matrix. And that's where the Krebs cycle takes place. And then the ETC, the electron transport chain, this is the last step in cellular respiration. And that literally happens on the inner membrane. So this membrane here, that is where the ETC is located, okay, is on that inner membrane. And that is where all of the different steps involved in cellular respiration take place. So like I said, the first step is glycolysis. And we're going to analyze this diagram here, and really we're focusing on what is going in and what is going out of this reaction. So in terms of things that go in, if you look on the far left here, we have glucose. That's what we start the reaction with. So this is our reaction, or what's going into the reaction. And remember, glucose is C6H12O6. And in this diagram specifically, that 6 represents the 6 carbons. And that's what's being represented here. A lot of these diagrams do what's called carbon counting. So it's not showing the complete molecule. It's just showing just the carbons. So, because if this diagram also had the hydrogens and the oxygens here, it would just be very busy and even more confusing. And then we are also going to invest 2 ATP. We kind of have to invest energy to make energy to split apart that glucose. And then the things that we're going to get out of this are 3 things. You can see coming out of that we have NADH, which is our hydrogen or electron carrier. H stands for hydrogen. Our 2 pyruvates, which are going to be used in the next step. And then we get 4 ATP. Now remember, we put 2 ATP in and we got 4 ATP out. So it's kind of like if you invest $2, or like let's say you spend $2 on a lottery ticket, and you win $4, you really only made $2 or 2 ATP, and this is what we call our net total. And that is glycolysis. Now from here, one of 2 things can happen. Again, like I said, this is always the first step in cellular respiration. but it depends if we have oxygen present or not. If we have no oxygen present, then we're going to stay in the cytoplasm and we're going to have fermentation take place. And this is also anaerobic because literally anaerobic means no oxygen. So this happens when there's no oxygen present. So when we have, like, let's say anaerobic conditions, we'll have fermentation take place. And this is different between organisms. In animals, like us, we will have lactic acid fermentation take place. And this is when, like, you're working out and your muscles are not getting oxygen fast enough. You're going to start to make that lactic acid, and that's that burning sensation that you feel. But as soon as you're done working out, you get oxygen back to your muscles. That goes away because that reaction is reversible. In plants, yeast, which is a unicellular organism, and bacteria, also unicellular organism, are going to do alcoholic fermentation. And this is how alcohol is made. You get ethyl alcohol as a product, and you also get carbon dioxide as a byproduct as well. Now, if you do have oxygen present, that will be aerobic, and you're going to move on to the next step, which would be pyruvic acid conversion. I'm actually we're gonna draw an arrow to this right here. This would be, so this is kind of like a baby step. So generally speaking, we do glycolysis, Krebs cycle, and then the ETC. And pyruvic acid conversion is like this baby step in between glycolysis and the Krebs cycle. So pyruvic acid conversion, we're going to take the pyruvic acid or the pyruvate. This is also pyruvate, that means the same thing. And we're going to use coenzyme A to change it into acetyl. And in that process, we get carbon dioxide and NADH. Carbon dioxide is a byproduct or waste product that you exhale. And NADH is one of our hydrogen or electron carriers. We already made some here. And we're going to use those later in the ETC or the electron transport chain. For this particular diagram of the Krebs cycle, which is what we're going to do next after we have converted that pyruvate into, or the pyruvic acid into acetyl, that was pyruvic acid conversion. Now we're going to enter the Krebs cycle. And for this particular diagram, you go around 2 times. So that acetyl is going to combine with the oxaloacetic acid. So the things going into this are the acetyl. Remember this CoA, it's just dropping off the acetyl. Okay, it's not actually involved in the reaction, it's just dropping it off at the Krebs cycle after it made it, converted it from pyruvate into acetyl, and then it just goes back to get more, converts it, and brings it back. So we have our acetyl that's going in. Okay, and we're going to kind of do a little, again, baby step here. It's going to combine with the oxaloacetic acid, and that's going to make citric acid. And then from here, we're going to get the rest of our products. So these are kind of like those steps in between. So our acetyl combined with oxaloacetic acid to make citric acid, And then from there, we break down that citric acid and we get a lot of different things out of this. We're going to get carbon dioxide, which is our byproduct or waste product. We just exhale that. More NADH, which is our hydrogen or electron carrier. We're going to get a little bit more ATP. We actually get 2 ATP because if you think about it, there's 1 here. You go around twice. 1 times 2 is 2. And then we also get our other hydrogen or electron carrier, which is FADH2. And then to continue this cycle, oxaloacetic acid, we end with that again, which is what we started with. This gets regenerated. And so up until this point, we've made quite a bit of NADH. We made some here in this step, and also this guy, which is another thing similar to NADH. We made it here, and we made it in glycolysis. So now in the last step, the ETC, that's where we're going to use that NADH. So we're going to use that NADH and the FADH2. They are going to drop off those hydrogens. And those hydrogens are going to split. We're going to get the hydrogen and the electrons. You can see that here in the diagram. And the electrons are going to power the ETC, okay? Specifically, this right here. So this is like the main part of the ETC. So this is powered by the electrons, and electrons actually have a lot of energy. And this is going to actively pump that H plus into this space. So you're going to put a lot of H plus into this space. Now that H plus now needs to go somewhere. So we have this very high concentration of H plus here. All these H's are going to leave through one exit. And again, this is happening, this is right here, this is that inner membrane we were talking about, and this is the matrix right here, the most interior part of the mitochondria. We're just very zoomed in. The process of those hydrogens moving across, going from that high to low concentration, is called chemiosmosis. And that's a type of passive transport. And they're going to move through ATP synthase. And as that happens, you're going to have 34 ATP made, which is quite a lot of ATP. So we're going to get 34 ATP. Now, all of those electrons that went on the ETC, they're going to be picked up at the end here by oxygen. So we're also using our oxygen here, and it's going to pick up the hydrogen and the electrons here that already went on the ETC. The hydrogens that cross through ATP synthase, and then those electrons that went on the electron transport chain. And that's going to make our last byproduct or waste product, which is water. So altogether, when all is said and done, you made 34 ATP in the ETC. You made 2 in the Krebs cycle, a net total of 2 in glycolysis, giving us a net total of 38 ATP. And again, this was all from taking glucose, C6H12O6, combining it with oxygen. There are 6 oxygens that are used. We get carbon dioxide and water as byproducts. And then, of course, our ATP or our energy that we can use to do things in our bodies.